This is lecture 33. It's part 6 of 7 of anabolic signaling. We're going to start getting into some applications. But as we've been doing, we'll pick up where we left off in the last lecture with some summarizing information. So this is a narrative summary of how and why mTOR gets turned on. The assumption of nutrition, that's the only part that's kind of complicated. But for example, normally insulin and food are inextricably linked. The invention of the hypodermic needle and the discovery of insulin, that's what created a divergence for that particular, in like the jazz age, for that example. And here's a look at a lot of mTOR regulating compounds we've talked about, segregated into different classes. So there's the immune system and other chemical activators and inhibitors. There's hormones, there's nutrition, there's mechanical sensors, and then there's drugs and supplements. And we'll get into that in the next lecture. Now remember that AMPK is the major enzymatic enemy of anabolism. And just like mTOR, AMPK has comprehensive regulatory pathways. So this is just a glimpse at some of those pathways, things that either activate or inhibit AMPK. And then this is a look at what AMPK is doing once it's turned on. There's lots of categories: fat metabolism, carbohydrate metabolism, protein metabolism. In this diagram, it looks like AMPK is inhibiting fat metabolism. This is a complicated relationship, which you don't need to know, but the article to the right here discusses it. It depends what tissue you're in. In skeletal muscle, AMPK is promoting fat oxidation. But the relationship between AMPK, hormone sensitive lipase, and lipolysis in actual fat cells, that's a little more complicated. Here's a passage from the same article explaining some of the findings. It depends what tissue you're in. And what the other, chemical and nutritional conditions are. But PKA and AMPK, these can oppose each other on hormone-sensitive lipase. But think of AMPK in general as catabolic to carbs, proteins, and fats. AMPK's protein catabolism, that's a bit clearer. Certainly simpler. And so here's a look at its inhibition of synthesis on the left and its promotion of degradation on the right. Okay, let's get into some mTOR-related practical applications and other considerations. We'll do non-nutritional applications today, everything other than food and supplements and drugs, and then we'll do that stuff in the next lecture. But as you would expect, the inhibition of mTOR is going to associate with a longer lifespan. Now, this is, it's usually being evaluated in yeast and mice, but it's not unreasonable to assume that the human form would behave similarly. Uh, so, with some amount of caution, not that much, but just a little bit of caution, we can assume that an excess of mTOR signaling would correspond to a shorter life. And then the inverse would obviously be true too. A shortage of mTOR signaling would correspond to a longer life. But it'd be like a long life devoid of strength and power and muscularity and expressions of athleticism that involve strength and power and muscle mass. So like nobody's going to choose you first in the kickball team, but you'll give the eulogies of everyone else on the field. So here's a look at the mechanism. We've talked about everything on this slide, so there should be some comprehension of what's being shown. If you knock out mTOR with rapamycin intermittently, you're inhibiting pathways that would reduce lifespan, and you're promoting pathways that would prolong lifespan. But chronic use of rapamycin, that could present some problems, as it inhibits the assembly of complex 2. All right, getting into long-term regulation of blood pressure. We have the renin-angiotensin-aldosterone system. So when pressure in the kidneys gets low, you don't need to know this. I'm just going to go through it really quick. When pressure in the kidneys is low. The kidneys squirt out renin into circulation, and renin is an enzyme. And it converts angiotensinogen, which is released by the liver, to angiotensin 1. And then ACE, angiotensin converting enzyme, it's another enzyme, obviously, that converts angiotensin 1 into angiotensin 2. Then angiotensin 2 vasoconstricts, then activates aldosterone. Aldosterone increases blood volume. So long-term regulation of blood pressure, there's a big kidney function here. And AMPK is a regulator of this. Activation of AMPK has a blood pressure lowering effect. So as we work our way through the lifespan, and hypertension gradually creeps up on us, AMPK might be increasingly friendly in this regard, helping us maintain blood pressure. And then looking at the heart itself, rapamycin treatment has shown to improve cardiac function um, and structure, both. So, like helping remodel left ventricular hypertrophy. And then one mechanism is inhibition of angiotensin 2, the elevations in protein synthesis in cardiac myocytes that would be caused by angiotensin 2. Okay, moving on to glucose regulation. We know how AMPK gets activated. That's what we're looking at here. And we know what PKB is, right? AKT or PKB, those are the same thing. And PKB inhibits AMPK. GSK3 does play a role in its inhibition, so you see that there. But zooming out, we see different pathways of GLUT4 translocation. So there's insulin-dependent mobilization of GLUT4. That goes through PKB, PI3K, PKB. But that also stimulates glycogen synthase. So you make glycogen out of the sugar that you bring into the cell. Conversely, non-insulin dependent glucose uptake, that will occur through AMPK activation. But that doesn't go through PKB, and it has the opposite effect on glycogen synthase. AMPK inhibits glycogen synthase, so you don't make glycogen out of the sugar that you bring into the cell. You consume it by activating glycolysis. So AMPK activates both hexokinase and phosphofructokinase. So AMPK brings sugar into the cell and burns it, while insulin signaling will bring sugar into the cell and store it. With AMPK activity or insulin signaling, early tissue healing might be enhanced owing to an increase in VEGF, vascular endothelial growth factor, and subsequent angiogenesis, just meaning you're going to have an improved blood supply. So in this study, they're looking at wound healing in diabetic mice compared to controls. And in the figure on the left, you see wound closure over 2 weeks. And controls heal the best, right? The ones that don't have diabetes. But metformin accelerates healing in the diabetic mice. And then in the figure on the right, the bar graphs, you can see angiogenesis. So differences in the early restoration of blood, which means the tissues would have access to nutrients. And metformin helped here too. Again, not as good as controls, but better than diabetic mice that weren't on metformin. But in the presence of augmented AMPK activity, terminal healing is likely to be compromised depending on the injury, because AMPK is turning off tissue remodeling and is turning on autophagy. And depending on the nature and location of the injury, you might induce apoptosis as well. That's what we're looking at here. It takes a couple of days to get going, but the difference in apoptosis between the control group and the metformin group was substantial. These are the same results as you just saw on the previous slide. This is just written out. This is a narrative form of what we just discussed. All right, our next topic is stress. and the stress hormone cortisol, which is a catabolic steroid. It's a glucocorticoid hormone. And remember, we have different muscle fiber types. We have type 1 fibers and type 2 fibers. The type 1 fibers aren't as sensitive to cortisol-induced atrophy. Cortisol has a much more robust effect on type 2 fibers, uh, which implies a relationship between chronic stress and strength and muscle remodeling and risk of injury and all of that. So this paragraph here, which explains the mechanism, should make sense to you. There's really only one unfamiliar thing here, which so cortisol upregulates something called REDD1, and then this red one promotes the assembly of tuberin. And if you're assembling more tuberin, that means mTOR is getting inhibited. So cortisol does have an inhibition of mTOR through that pathway. And then cortisol also upregulates FOXO, which, as you know, promotes proteolysis, both through the lysosomal and ubiquitin proteasome systems. And then cortisol also, another also here, promotes the transcription of a protein called KLF15. And then KLF15 promotes MURF1 expression. And then MRF1 contributes to the ubiquitination of myosin heavy chain. So it's selective muscle protein degradation. And then something else that ALF15 does is regulate BCAA branched chain amino acid degradation, and leucine is one of your branched chain amino acids. So leucine is affected here. You'll see a reduction in leucine content, and subsequently you'll see a reduction in mTOR signaling. And then our last point with cortisol is this relationship with testosterone. It's an adversarial relationship. Elevated cortisol negatively impacts circulating testosterone. So by a bunch of mechanisms, a number of mechanisms, cortisol is anabolic and pro-catabolic, targeting leucine and myosin, and especially the type 2 muscle fibers. All right, so now let's look at resistance training. This article measures cellular hypertrophic signaling in different muscle actions in a rat limb muscle, but it's looking at MAPK signaling specifically. So it compares nothing to passive stretching, to concentric loading, to isometric loading, to eccentric loading or eccentric loading. Um, Stretching doesn't do anything, right? Concentric is barely better than that, barely better than stretching. Isometric, that's getting pretty good, and the eccentric is king and queen, um, and all the regents and whatever. So this is what we would expect to find, though, knowing that eccentric stress corresponds to more disruption of the sarcolemma, where arachidonic acid lives. And then also, so this is just a narrative version of that visual representation that we just looked at. So again, eccentric is best, signals MAPK the most. Isometric, second best. Concentric, second worst. And passive stretching is the worst for that particular signaling. Now, you also know that lots of cytokines will induce a hypertrophic response, things like interleukin 2, interleukin 15, and we expect a bigger immune chemical response after eccentric loading. More tissue disruption. We're going to see more IL-15, and that IL-15 is going to help us with more PI3K signaling. We also have stretch activated channels. We talked about these. These permit the influx of calcium into the cell. And eccentric actions, more than concentric, are going to stimulate the stretch activated channels. And then intracellular calcium appears to activate mTOR, assist in the activation of mTOR. So if you introduce SAC inhibitors, stretch activated channel inhibitors, that will attenuate PKB activation. So this is another explanation for increased mTOR signaling following eccentric muscle actions. And it's been seen in rodents and humans alike, that one. Okay, so mechanogrowth factor, MGF. This is a variant of IGF. It's released from working muscle cells and it acts locally. But exercise-induced muscle damage is a trigger for the increase in MGF levels. And eccentric stress is what causes the damage. So it's no surprise that eccentric stress induces more MGF, which causes autocrine signaling of both PI3K and MAPK. Although IGF does go through PI3K more than MAPK, it does signal both. Same article, but now we're looking at the suppression of myostatin. So eccentric activation of the tissue suppresses myostatin the most compared to isometric and concentric. And we know that myostatin, it's a myokine, a little protein released by the muscle, inhibits PKB. So with eccentric activation of muscle, the suppression of myostatin would permit more PKB signaling. All right, moving on to integrin signaling. This is especially sensitive to eccentric loading as well, and it's a major signal for hypertrophy following exercise. In this study, they found eccentric activations activate focal adhesion kinase FAK significantly better throughout the duration of the study. Looking at the results, whether they're looking at the total phosphorylated FAK levels or a phosphorylated to total FAK ratio, eccentric exercise stress was much more effective at relaying anabolic signaling through the integrins. Um, and then the authors, they mention here vinculin. This is a linkage protein that anchors integrin to the actin cytoskeleton. You don't need to know what it is. It's just they mention it, so I figured I would explain it. But okay, remember that myonuclei are critical to growth, to hypertrophy, the growth of the muscle cell, and they're donated by satellite cells. That's what the SCs are in the text here. And the accumulation of new myonuclei, that's what permits the growth of the muscle. And eccentric stress corresponds to more satellite cell activation, especially in the type 2 fibers. Okay, getting into the speed of your contractions, there may be reason to take your repetitions a bit slower, limited data, but in this study, they found slow eccentric stress generated more MGF, mechanogrowth factor, than the fast eccentric stresses. The slow was 30 degrees per second, compared to fast, that was 210 degrees per second. All right, let's talk about size principle. First off, mechanotransduction is going to belong to the fibers that were actually activated. Passive fibers aren't really participating in the sensation of and response to the application of mechanical loads. So there might be a little bit of like a neighborhood effect. So like, do adjacent muscle cells benefit from neighboring cell activation? Yeah, sure, a little. There are transmembrane proteins that sense community activities, but it's not as important to enhance tissue that was totally uninvolved. The cells that bear the bulk of the responsibility reap the bulk of the rewards. Muscle metabolism isn't really a socialist economy. Fitness has to be met by reinforcing the cells and tissues that are most threatened. And if there's a little bit of spillover, great. The community sees improvement, but the individual reaps the greatest reward. So in this tug of war, in this picture, this little, tug of war, the girl in the galaxy pants, she's not pulling. I mean, she's almost leaning forward, so she has no business sharing in the trophy if her team wins. And if she were a muscle cell, pulling on a bone, and it wasn't activated, that muscle cell wasn't activated during the exercise session, just part of a high threshold motor unit or whatever that was never called on, her mechanosensors would be dormant. So it's important to activate muscle cells, whichever ones that you want to respond to your exercise. And so how would you do that? How would you activate the type 2's high threshold motor units? Ascend Henneman's ladder. And the most reliable way to do that is with a heavy load. And then also hormonal and chemical signaling. These are facilitated by cellular recruitment. A lot of that is accounted for by autocrine effects, but Hormonal, chemical, and mechanical signaling are all facilitated by use, by activation of cells. And on the subject of climbing Henneman's ladder, it's best to do it with a high load as opposed to a prolonged time under tension, because mere activation of a motor unit isn't sufficient to induce hypertrophic signaling. Greater mechanical intensity seems necessary as the type 2 fibers are more susceptible to mTOR signaling than type 1s. It's just the phosphorylation of P70S6K is going to be less dramatic in your type 1 fibers. So how do you climb the ladder? That's going to matter, right? Incorporating heavy loads is the way that's going to be most effective as opposed to just holding the weight forever. And then exercise volume matters too. It's not just load. The amount of muscle mass involved, the intensity of exercise, and the duration of your exercise, all of this is going to affect the amount of testosterone and IGF you see in circulation. And if lower intensity resistance training, if that's all you can do, if this is combined with reductions in blood flow, to the working muscle, through vascular occlusion, P70S6K is going to be activated more, and protein synthesis will be improved more. So if you can't lift, heavy, if you can't, climb up Henneman's ladder in a heavy way, you can include blood flow as an alternative. That lower oxygen is going to correspond to a higher reliance on glycolysis and glycolytic fibers, and you'll see increased markers of exercise intensity like lactate without performing the intense action, the heavy, at least, action. So you sort of fool your body into thinking you've exerted yourself into an anoxic state, and you'll see increases in growth hormone. And in this study, 3 hours after exercise, there was an increase in P70S6K phosphorylation in the inclusion group. But you see lactate and growth hormone, these both being increased. Plenty of studies on this. Here's another. I'll show you a summary of the findings in just a second. But there are a number of mechanisms that could be present. I mean, one of them is you get a pump, right? You do a better job of occluding exiting blood than blood that's entering. And so swelling, which you'll experience, that can activate anabolic signaling, both FAK, focal adhesion kinase, and MAPK. And they did find MAPK signaling. They didn't find any difference in FAK activity, though. Nor did they find any difference in AMPK activity in this study. But here are the findings. There was a 9-fold increase in peak growth hormone levels after exercise in the blood flow restriction group. And then they saw activations of both PI3K mTOR and MAPK. Here's a visual representation of the findings. You do see cortisol elevated too, and we already discussed about how cortisol has catabolic effects. In the next lecture, we'll talk about ways to mitigate that specific thing, that specific effect. But they did find differences in mTOR, P70S6K, and MAPK, ERK, phosphorylation. And so greater activity in the blood flow restriction group in all of those. Some contraindications, though. So, like if you have diabetes or cancer or compromised renal function, I hold off on blood flow restriction or hypertension. Depending how hyper, if it's just barely, there's gradations here. But like if you have sickle cell or a history of DVTs, some sort of altered cardiovascular function, I would avoid incorporating this into your exercise. And maybe if you're on a medication that could increase clotting, that's another reason. That's another contraindication, another reason to avoid blood flow restriction. Okay, something else to acknowledge about resistance training is its effect on total energy expenditure. Skeletal muscle, protein turnover. This is a large part of the body's total metabolism. Maybe a fifth, something like that, of your resting metabolism. And then when muscle is growing, it does so with great expense. Protein synthesis accounts for an enormous percentage of a cell's like total energy consumption. It's depending on the creature. There's lots of studies that talk about this. I mean, it depends on what animal we're looking at, what organism we're looking at, and then what cell we're in. But translation is the bulk of that cell's expense, and then microbes, protein synthesis is three-fourths of the total economy. Here's a general hierarchy of metabolic costs. Protein synthesis above all else. I mean, this is really expensive, translating protein. So if weight loss is your goal, protein translation is a great way to burn calories. Okay, let's talk about the timing of adaptations. Early adaptation, this is mostly neural. Later is when you start getting into hypertrophy. At the onset of exercise, those first couple of weeks, you will see a lot of protein synthesis, muscle protein synthesis, but it's not aimed at hypertrophy. It's aimed at repairing the damage that you're doing. So think, by way of analogy, if like a rogue earthquake, it's just some rare event damages your house like once, you're probably not going to build that house back, bigger and better than ever before, but you'll fix it, right? You'll repair it back to baseline. But if those earthquakes become a consistent phenomenon, you're just regularly experiencing them, you'll probably commit more resources to the remodel, reinforcing your house so that it can tolerate future quakes. So you do make it, bigger and better and more expensive and whatever than before, but not right away, only after a lot of exposures. And the same is true with skeletal muscle. You don't hypertrophy right away. Now, maybe you'll see cross-sectional area increases pretty close to the start of exercise. But the differences you do see are mostly muscle swelling, right, from the damage. We talked all about swelling in the second block, and that accounts for most of the cross-sectional area differences early in exercise. And then there may, in addition to that, be some connective tissue remodeling that can account for a little bit of that size change. But real muscle hypertrophy, this isn't going to be detectable until you've been at it for at least a month, probably a month and a half. And then depending on the training you do, there are differences in the genetic response. So what proteins are you translating? Are you assembling an oxidative phenotype, or are you accumulating skeletal muscle proteins and increasing cross-sectional area for enhanced strength and power? At the onset of an exercise program, a beginner can make progress in both directions. Beginners can become better at oxidative tasks and better at strength performance. But the more they exercise, transitioning from beginner to novice to intermediate to advanced, they get worse and worse at versatility. So let me read the end here. I'm quoting. It is only with sustained exercise training that there is a fine tuning of the transcriptome, the protein synthetic response, and then the proteasome that gives rise to divergent hypertrophic and oxidative phenotypes. So someone with some experience isn't going to respond as well to mixed signals. We align with a phenotype and begin to defend it. So when you do aerobic training, you can experience an increase in protein synthesis among genes relevant to aerobic performance for a number of hours, but it's not going to carry into the next day. For resistance training, after performing it, you're going to see mTOR turned on for a dozen or more hours after you re-rack your last weight. And you'll be translating protein for a lot longer than, I mean, especially if the workout is something that gets you sore for the next 3 days. So muscle protein synthesis is not a briefly transient phenomenon after resistance training, but after cardiovascular activity, it doesn't last too long. Um, and now that you know how long mTOR can be active after a workout, consider what the inclusion of an aerobic component after your resistance training might do. So in one session, right, you lift weights and then you follow up with some cardio. I mean, if you want to get good at cardiovascular endurance, go for it, right? The resistance training doesn't really mess up aerobic performance much. But cardiovascular training will impair the anabolic signals, the anabolic responses to your exercise. And you know the signaling cascade, you know everything on this diagram. The anabolic agents that are released with resistance training are inhibiting tuberin, they're activating raptor, and then the AMPK that gets activated with aerobic stress is activating tuberin and inhibiting raptor. So you're getting contradictory signals with sort of an anabolic resistance training program and aerobic stress. Now, sometimes people will claim that there's no impairment in anabolic signaling in the presence of aerobic stress. And if they cite anything at all, they tend to cite these articles, which looked at concurrent strength and endurance training and found sort of that maybe it's not as damning as others have found it to be. Again, maybe, but so let's look at the actual articles. We'll start with this one. This is out of Finland, which William Kramer's group worked with. And he's an author on this article. And none of the subjects in this study had a history of resistance training. So it doesn't really matter what gets added with it. As we discussed just barely earlier in this lecture, initial responses are different from the responses of someone more experienced who has developed phenotypic traits already. So you can expect novice resistance training to do something in these, novices. Um, and so they had them exercise for 21 weeks, plenty of time, 21 weeks. Either just resistance training, 2 days a week, or resistance training with a couple of aerobic training days on top of it, or riding a stationary bike for a while. Specifically measured while, 45 minutes, 60 minutes. It depends at what point in the study they were in. And then this graph shows, this is one of the findings, the difference between the strength group, marked S on the graph, that, again, 2 days a week of resistance training. S compared to the strength and endurance group, SE, on the graph. So 2 days a week of resistance training and 2 days a week of endurance training. And so the maximal rate of force development, RFD, on the graph shows the strength only group to have improved much more over the 21 week study period. Um, and I mean, although other muscular differences weren't really observed in these, beginners and these novices, this is hardly evidence that aerobic training doesn't impair resistance improvements. And then this study used 10 college students, all males, recreationally active, no lifters, and they did 5 weeks, just 5 weeks of a one-legged training. So there's 45 minutes of one-legged cycle ergometer, one-legged exercise bike, and or, depending on the leg, one-legged leg extensions. And then they found that the aerobic leg did better overall. But again, we have a bunch of people who don't really exercise doing certainly don't lift, doing either more or less total exercise with each leg for 5 weeks. So we would expect people to make progress in both directions in this short of a study. But beyond that, if you do like let's say you're doing only bicep curls and tries only resistance training with your arms, but you're doing tons of aerobics with your legs, your arms still don't grow that well. Because they're all connected to the same body, right? So the one leg versus the other leg study design doesn't really make sense. So you don't really have to read past the methods in this article. So another mechanism, in addition to AMPK impairing mTOR signaling, is testosterone. Performing a high volume of aerobic exercise is associated with reductions in testosterone. The condition even has a name, the exercise hypogonadal male condition. And just as expected with low testosterone caused by anything, the subjects tend to experience symptoms. And then the abbreviations on this slide, AMS, that's aging male syndrome, and BMD, that's bone mineral density. Okay, let's talk about specificity of adaptation. That's the topic we concluded block one with. Mechanotransduction operates according to the principle of specificity too. The different types of forces endured imprint differently on the cellular signaling processes. So think about the mechanical forces that any cell would be responding to. So let's take epithelial cells, what we're looking at here. How the blood flows through your vessels matters, right? Whether it's laminar, like smooth flow, or oscillatory, like rough flow, that imposes different mechanical forces, and this results in different biochemical responses. And then in a myotube, which is a developing skeletal muscle fiber, there are differences in signaling cascades in response to uniaxial stretching or multiaxial stretching. And that C2, C12, you see, that's an immortalized mouse myoblast cell line. You see that in in research. But the point is mechanoreceptors sense a lot. They sense specific qualities of the mechanical stimuli, and then the adaptations that follow are just as precise. All right, so how do we apply this to weight room strategies? I mean, the other components of the fit principle, in addition to type, those matter too. Frequency, intensity, and the duration. Um, all of these affect signaling responses. And just activating the motor unit, that's not enough, right? The load matters, the timing matters, the characteristics of the exercise, concentric versus eccentric, all of this matters in signaling. And then angular specificity, we talked about this earlier in the semester. If your elbow flexors, you hold them at 90 degrees, and you can expect improvement from maybe 70 degrees to 110 degrees. But the most of your adaptation is going to be at exactly 90 degrees, less at 95, less at 100, less at 110, and then sort of no responses after that. So that's angular specificity. The mechanical signaling of any isometric hold um, is going to be different from the signaling induced by dynamic exercise, moving throughout a range of motion. Just as concentric and eccentric actions elicit different adaptations, so does isometric. At every different angle, there's going to be a different adaptation. Just depends where you're holding it. Our sensors, our mechanosensors are sensitive. They relay specific information, and our adaptations are precise to that specific information. All right, and you remember the Alfredson protocol. If all you do is stretch, this signal will probably be filtered out. But if there's an increase in tension, an increase in load at the same time, the mechanical signal is amplified. And this is important because tendons don't have inflammation. They really rely on mechanical signaling, and the Alfredson protocol is a way of getting those mechanical signals to fire better, to fire more appropriately. And yeah, in this case, make the Achilles more responsive to the signals for repair and remodeling. And the overall application here is illustrated well by Davis, Wolf, Agassi, and Tyson. Translating mTOR to the clinical or the sporting context, if you know what outcome you want to achieve, and you know the cell signaling map, you'll know how to navigate from point A to point B. So start with a task analysis. Figure out what traits you want to bolster, what capacities you want to develop, and then just plot your journey through the signaling cascades, and you'll arrive very effectively. But that's it for this lecture. Here are the review questions, and I'll see you in the next one where we talk about applications involving nutrition and drugs and supplements.